We are thrilled to announce our participation in the upcoming Professional Association of Teachers of Students with Specific Learning Disabilities 2024 Annual Conference. This event presents an excellent opportunity to explore how neuroscientific insights can directly and indirectly shape our approaches to supporting students who are struggling with reading. This is much more than just a conference. It's a gateway to pioneering educational practices that promise to enhance teaching methods and enrich our students' learning experiences.
We will be coming to you from Teachers College, Columbia University, and the Neurocognition of Language Lab in New York City. Our lab conducts research in the neural basis of reading and its implications for teaching and learning.
So I'm Karen Froud. I'm the director of the Neurocognition of Language Lab, and I'm a professor of neuroscience and education in the Department of Biobehavioral Sciences at Teachers College, Columbia University, right here in New York. I trained as a speech-language pathologist originally in the UK and conducted research at University College London and Massachusetts Institute of Technology before joining the faculty here in 2003. And since then, we've been conducting brain imaging research on populations across the lifespan with a focus on clinical and classroom questions and practices. I'm very lucky to have a fantastic team of researchers and educators here to facilitate that work, and my close colleague, Dr. Lisa Levinson, will also be joining me for this presentation.
I'm actually a full-time lecturer in the Neuroscience and Education program, and I also collaborate in the Neurocognition of Language Lab here at TC. The emphasis of my research is focused on reading. And I'll also share that I bring a unique perspective in that I myself have dyslexia.
And the lab conducts theoretically grounded empirical research using behavioral methods and electroencephalographic methods, which we'll talk more about in just a second, to increase understanding of the processes and representations involved in speech, language, and cognition, and to inform best practices in clinical and educational venues.
The research we do primarily involves electroencephalography, or EEG, which records the brain's activity using nets that are fitted with electrodes embedded in sponges that sit on the scalp. These electrodes pick up the brain's electrical signals, which need to be amplified for analysis. The process involves filtering out noise or unwanted signal to isolate the brain's activity related to a particular task. The amplified and processed signals are then converted to a visual waveform. These waveforms represent the brain's electrical activity over time, allowing researchers to index responses to timed, locked stimuli. The entire process is non-invasive, safe, and painless, providing a window into the brain's functioning through its electrical chatter.
Right, and we can use this method to capture information about what happens in the brain during complex cognitive tasks like reading. In fact, all of our experimental inquiries require us to synthesize the broader research, and we attend and host scientific conferences related to the science of reading. So this puts us in an excellent position to share the neuroscientific perspective on reading acquisition and the challenges that can present and the implications for classroom practice. We are working hard to push educational neuroscience into the classroom, and to that end, we do whatever we can to foster bidirectional relationships with teachers who are dealing firsthand with the individual differences that define learning.
At the heart of our mission is the empowerment of educators with innovative tools and insights derived from educational neuroscience. This perspective is crucial for understanding the challenges and diverse needs inherent in learning differences. Our work aims to bridge the gap between neuroscience and classroom practice, bringing cutting-edge neuroscientific understanding directly to teachers who can apply it most effectively. We are committed to fostering a bidirectional relationship with educators, recognizing that their firsthand experiences with individual learning differences are invaluable in shaping and refining our approach. Through this collaborative effort, we aim to integrate educational neuroscience into everyday teaching, enriching the learning experience for all students, especially those with learning differences.
So we can't wait to get together and discuss how does the brain form the necessary networks and the efficient circuitry to support reading, and how do evidence-based strategies contribute We hope you will join us for a stimulating and engaging presentation on the underlying neural mechanisms and their impacts on learning.